Hello friends, welcome to TECHQUEST channel. This is for the medical technologist by your tech. In this video, we will discuss about the CSF cell count. I just listed 10 tips for accurate counting of CSF samples. Sample handling and delay in sample processing. Delays in processing of the CSF sample or improper storage can also affect cell count as cells can get degenerated or clumps over the time. This can lead to inaccurate cell count and affect the interpretation of the results. Prompt processing of CSF samples are crucial. If delays are unavoidable, appropriate preservatives or protein supplements like HETA starch or fetal cough serum can be added to maintain the cellular integrity. CSF sample often contains very low numbers of cells, making accurate count is challenging. Inaccurate counts in this range can have significant clinical implications for diagnosis and treatment decisions. 2. Importance of thorough mixing. Inadequate mixing of the CSF sample before analysis can lead to inaccurate cell counts as cells may settle or clump, especially in low volume samples. This can result in underestimation of the true cell count or inaccurate differential count. Gentle but thorough mixing of the sample is essential before any cell count. A vigorous mixing can lyse the RBCs and degrade the WBC morphology. 3. Counting chamber cleaning. Sometimes the oil on the oil immersion may touch the counting chamber cover slip. These oil drops can obscure the vision, cause inaccurate cell count. Handle the counting chamber and cover slip with care to avoid scratching or damaging the grid. Remove the cover slip and clean both the counting chamber and the cover slip with lens paper or soft tissue. Wash with water to remove any residual CSF or debris. If necessary, use a mild cleaning solution like 10% bleach solution. Alternatively, a diluted ethanol solution can be used to clean the chamber. Thoroughly dry the chamber and the cover slip with a soft cloth or allow them to air dry. Residue of water droplets or cleaning solution can lyse the blood cells. 4. The cover slip thickness matters. The standard size for CSF counting cover slip is 20 into 26 mm, though larger sizes are available. The thickness of the cover slip should be 0.4 mm. The thin cover slips used for slide microscopy cannot be used as thin cover slips can bend and give inaccurate cell count. 0.4 mm cover slips are ground and polished to ensure a flat and even surface, as these are used to cover the counting chamber, limiting the volume of the CSF sample and ensuring precise cell count. 5. Charging the CSF onto the counting chamber. A small drop of CSF or diluted sample is introduced to the chamber, allowing capillary action to fill the space between the cover slip and the counting grid. Microhematocrit capillary tubes can be used to transfer the sample to the chamber. Make sure the sample is spread completely on the chamber. Never use a micropipette to inject the sample to the chamber, as it can overcharge or allow the cover slip to float. The amount of sample present in the space between the cover slip and the counting grid is important for the accuracy of the sample results. 6. Settling of cells. The counting chamber with the sample is placed in a petri dish used in microbiology for 3 to 5 minutes to settle the cells. A small amount of wet cotton or wet tissue paper is placed near the counting chamber and closed. The cells can move and give inaccurate counting if the cells are not settled. Number 7. L rule counting. When cells are present at the edge of the square, it may be confused to count them or not. The L rule provides a clear guideline. Count cells touching the top and left lines square and ignore those touching the bottom and the right side of the lines. Applying this rule constantly ensures that each cell is counted only once. regardless of how many squares it might appear to be touching. L rule prevents overestimation of cell counts due to double counting. It ensures consistent cell counting across different users and experiments. L rule provides a reliable method for determining cell count. By adhering to the L rule, cell counts become more reliable and easier to reproduce. 8. Differentiation of RBCs and WBCs. In CSF, RBCs should not be ideally be present. WBCs should be present in very low number. The presence of RBCs in CSF can indicate bleeding or a traumatic tap, and increased WBC can suggest infection, inflammation, or other conditions affecting the central nervous system. The size of RBC and a small lymphocyte are at the same size. The cells are unstrained during counting. Many techs have difficulty in differentiating these cells. The easy way is the presence of refractile or shining of RBCs when using a fine adjustment in the microscope, or a drop of toluidine blue stain can stain the WBCs if you have a doubt. Number 9. Sample dilution. A dilution of the CSF sample is often necessary, especially if it contains RBCs. A 1 in 10 dilution is commonly starting point, but higher dilutions may be required if the cells are more. WBC diluting fluid may be used for WBC counting, as this can lyse the RBCs and give more accurate WBC count. Number 10. Calculations. The CSF manual cell count calculation is very important part for accuracy. The dilution made with the sample, the depth of the counting chamber are important parts of calculation. The area counted may be decided with the amount of cells present in the sample. If the sample contains more of cells, automated cell counters can be used for measuring CSF cell counts. The normal WBC count is below 10 cells and normal RBC count is 0. CSF samples should be analyzed within 1 hour of collection to minimize cell deterioration. Fluids older than 24 hours are generally not suitable for cell count due to potential cell lysis and degeneration. Thank you for watching. Please subscribe, like and share this video. Thank you.